Well, hello, everybody. Wanted to say hello and thank you for joining us. I'm Steve.
And I'm Dennis.
This is the Natural Growing Through Biology podcast series. And we normally, at least to date, have been focusing on some kind of cool functions of microbes, what they can do, what we expect them to see. But we also get a lot of questions about, well, tell me more about your products. What product should I use? How should I use that product? So we wanted to get into a little bit of that sort of information, a little bit on product selection, what products we have, how to use them, what successes we're seeing, and that sort of thing, how to get the most out of the products in your field. So we're going to talk a little bit about product today, aren't we, Dennis?
Yeah, it's gonna be a fun podcast series.
Sure.
It's not as I guess we kind of get really excited about all the different things microbes can do and kind of geek out on that a little bit because it is so cool.
And our products are so cool as well.
They really are. So let's get started and let's give you a little information about how you might want to use those products over the year.
Absolutely. So one of the first places that oftentimes we have grower starter, or a lot of times people kind of get into this, is kind of a pre-digestion. It's a primer, and we've heard it called the fall primer, the soil primer. But the idea here is to get those microbes established late in the season so that they're in the soil and functioning and digesting and breaking down and generating soil and extracting a lot of nutrition so that the following spring, during our following growing season, a lot of that nutrition, a lot of that groundwork has already been laid so those plants can have access to more nutrition. And the biology that we're going to then apply through In-furrow or some of our other application techniques, they've already got a bit of a leg up. So we have a lot of people using this fall or soil primer program to great success. And again, the benefit there is one of the hardest things. The beauty of biology is how functional it is, but the reality of biology is most of these organisms, they're like you and me. They get cold. They get kind of sleepy. And when the soil temperatures are not where we need them to be to have those microbes functioning at optimum, we don't get optimum performance out of it. Kind of a duh. Well, yeah, that makes sense. So one of the ways we overcome that is setting that stage, getting some of those microbes established with the plant or to help break down some of the field debris that was generated during the previous season. The way that we do that, again, is that fall primer program.
Yeah, and a lot of times we look at that based off of, I call it, Steve mentioned the pre-digestion, but we have that field debris out there. We want to break that field debris down, pre-digest that, get that ready for the next year's crop. Really what we're doing is we're composting in the field scenario. Yep. That's what we're doing. Yep. We're just increasing that microbial community around that field debris to break that down and pre-digest it. And you know, a lot of times like the guys back east, the non-GMO corn or the GMO corn, things like that are a little bit harder to break down. You know, a lot of times we look at that based off our biodigester program. You know, you put out the biodigester, it's designed to, as a cellulose digester, to break those things down in the field environment.
More debris means you have more to digest, so we go after it a little more aggressively.
Exactly. You know, a lot of the growers that may not have as much debris in their field environment are using our spectrum, and that's probably what is more known on the fall primer program is
most
that
common.
that you know, that spectrum application. You know, a lot of times I look at that from the standpoint of a half rate of spectrum with our nutrient, a little bit of fish as a food source for the microbe. We need that nitrogen to break that carbon chain. So that's really what the fall primer program is about. And you know, we kind of laugh because it's springtime, we're getting ready to plant. Why are we talking about the fall primer program? Because it's not really only just for the fall. A lot of the growers that I look at right now is we're getting ready to plant our trees, our blueberries, whatever it might be. A lot of times if we come in with the spring application also in furrow right where we're going to put those trees, we can start that digestive process a little bit early. And a lot of times like that, I look at like the OP8 product. We come in with the OP8, the nutrient. Now we're banding it into the furrow, targeting it right where that plant can actually use it, where we're going to put that plant. See that in a lot of the vegetable growers, row crop vegetable growers, those types of things can utilize those products. So when we move into the next step, which is a lot of times what we talk about, the in-furrow application, we've already pre-digest that. We've set up an environment not only for the plant, but also for this other microbial community to have more readily available food. Remember, everything is interconnected. That's what Bruce always used to talk about. Everything we do builds upon. It's that stair-step as Dr. Hatfield talks about. So if we can do that pre-digestion, we can do that fall primer or spring primer program, it just enhances the environment for everybody.
Oh, absolutely. And the goal here, we need to keep in mind the microbial carbon pump and the reality that we're seeing that 50% of our organic matter, according to research, is showing that it's dead microbial biomass. So we can be generating soil over the winter. It's not as fast, and a lot of people ask, well, it's too cold, it's frozen, those microbes aren't doing anything. Well, they are. They're slower, but as long as they have access to liquid water, they're going to continue growing and digesting and breaking things down. Again, it's just much slower. However, if they're already present and we've laid that foundation, then as soon as those soil temperatures start to climb, we've got a little bit of a leg up. And that leads us right into...
Yeah, well, and I was going to say, this is... Whether we have a cover crop out there, obviously, any time we have actively growing roots in the environment, it increases the diversity and the function of the microbial communities. But this also can happen on just field debris soils. We're just trying to break that down. The microbial strain may change a little bit based off who is most dominant within that soil environment, but we're still getting that digestion throughout the fall, into the winter, and then as those temperatures warm up the next spring. So it's really a great way to do it. But it really preps us for what we talk about is our seeding for that next year. What are we doing that next season? And, you know, I always get my favorite when I start to talk about row crop agriculture is the in-furrow application.
Oh, it's beautiful.
Yeah. If we have the ability of liquid application right over the seed, I'm not saying offset 2x2, whatever it may be, right over the top of that seed. We all talk about, you know, Dr. White's research on the rhizophagy cycle, talking about how that seed at the point of germination is putting out those exudates to recruit the microbial community for root production to be normal root production. And that's really what that's the foundation of a healthy plant, is that root structure and that biology that surrounds that root structure within that soil environment. So if we're doing an in-furrow application and that microbial community, that inoculum is right on top of that seed, that's right where we want it. The other thing I love about the in-furrow application is the idea we can put a support package in there. Not so much for the plant, but for that biological microbe food, that biological community. We can put a little bit of fish, a little bit of humate. We can put all of those microbe foods. So while we're in that transition period of germination, they have a sack lunch. They have everything that they need to get off and start. And this also helps with the germination of that seed.
Oh, absolutely. So again, the goal, like we talk about, most of these that we're talking about, they're plant growth promoting rhizobacteria, plant growth enhancing fungi. These are all organisms that nature has designed to go and live right there, literally around the root, on the root, in the root. In the case of the endophytes, we're creating that rhizo sheath. And we see some really incredible pictures when we have people doing some of this in-furrow, where we pull out a plant and we see it, and the plant emerged, germinated, 10 days ago, a week ago, and you've already got that full rhizo sheath. Those rasta roots are already developing. And the beauty there is we have already allowed that plant to access more moisture, to have better gas exchange, to have better nutrient access. pH shifts, if we're struggling with some pH shifts. That rhizo sheath, it's a magic zone. It is just like building a house around you. It protects you from the environment, it protects you from what else is going on, and it gives those plant roots the best possible access to all of those things that we often talk about. We need light, we need air, we need moisture, all of those things. This rhizo sheath is allowing that plant to have access to more of that, as well as creating a physical buffer and a physical barrier to other organisms that may be out in your soil. Because we all know sometimes that spring soil, that cool, wet system, it can be hard, it can be detrimental. So if we already have those beneficial organisms feeding the plant and improving the plant's health, we don't worry about it as much.
Yeah, and so a lot of times in springtime, seed inoculation, in-furrow, when we're doing a liquid application, a lot of times I'm always looking at our Spectrum Plus Myco. It has the broad diversity of beneficial bacteria plus those mycorrhizae fungi. And the key to that is in that those mycorrhizae spores need to come in contact with the root in order to create that relationship.
There's
based
communication.
Off a plant need.
There's communication that happens there.
Yeah. And so if that spore is not right there with the seed, there may be a delay of creating that relationship if that plant needs that relationship based off of phosphorus or whatever that initial response might be. So in-furrow right on top of the seed, the Spectrum Plus Myco. Again, we always talk about the Nutrinede, our enzyme product or our Pepzyme G. We'll talk a little bit about enzymes later here in the presentation and how important they really are over in not only plant health but also microbial communities within that soil environment.
Enzymes are what drive every biochemical process in our bodies, in a plant body, in a microbody. The reality is these enzymes, these specialized proteins, are what creates life. They're what allow life to happen, because it couldn't happen without these. And it's incredible what we can have happen when we start increasing the digestive capacity through some of those enzymes. Okay, sorry. Back to In-furrow.
That's exactly it. And so a lot of times Spectrum Plus Myco, one of the enzyme products with a little bit of fish, a little bit of food, is what we're recommending on our In-furrow product. And a lot of times we have growers that are using biological if they need a little nutrition package, something that can complement that biology based off of testing.
Wait, test don't guess?
Exactly. Plant sap analysis, soil analysis, understanding what our deficiencies are being expressed by that crop.
Yeah. And in those cases, we can target in those directions.
Absolutely. So basically to create overall plant health, more photosynthesis, feed those microbes within that soil environment. But a lot of times I look at, too, if we have a short-term crop, we're talking about our spinaches or our lettuce or something like that, very short-term crop that may not create that relationship with the mycorrhizae depending on delay. It doesn't have time. Then a lot of times we can look at one of the other biological inoculants based off of function.
Oh, PSB fits with those fantastically.
Yeah, so, a lot of the wheat and corn growers that I'm working with that are backing off on nitrogen are utilizing the Spectrum NFB as an in-furrow application. To get those nitrogen fixers, again, right down in that root zone around that plant so they can start fixing nitrogen, create that relationship with the plant. So a lot of times it's the understanding. Most often our recommendation is Spectrum Plus Myco, but there are a few variations there based off of function and exactly what we're trying to do. Because when it comes down to it, these biological inoculants provide specific functions based off of plant expression. The plant is communicating with those. So let's use the right inoculum designed for what we're trying to do within that crop.
If we have a salty environment, we're going to come in a targeted approach with our Spectrum DS. We're going to be suggesting extra calcium because we know that we're going to struggle with that, extra humate to help bind that up, some seaweed to help with that plant stress. So again, we have our general suggestions and our general protocols and programs. But if you have your own specific issues, reach out, talk to us. We'll give our honest opinion on what we think would be the best product, because that's our goal. Our goal is to get the best performance out for the growers so they can have the best quality soil, the best quality plants, the best yield, the best health. Because, the goal is to generate food that is life-giving. We want to create a commodity. We want to create a crop that's actually food for people. Isn't that a crazy concept?
That is a crazy concept. But, if we don't have the ability to do in-furrow treatment, we always look at the seed treatment. We have a lot of guys that are using the seed treatment. The difference between the in-furrow with the liquid application, the seed treatment, we're limited on the support and food that we can put around that with that biological inoculum at the time of planting, because we can only put so much on that seed. And depending on what type of seed we're looking at, some seeds actually will hold a little more material than others. So we always have to look at, okay, what is my seed size? What is its shape? How am I going to get this biological seed treatment to stick? But in most cases, we're looking, again, Spectrum Plus Myco as a seed treatment. A lot of times, we'll say mix that with like a humate powder, another sticking agent. When you're talking 155 grams per acre of the Spectrum Plus Myco, it's nice if we can build up that volume with microbe food in order to get a better coating onto that seed treatment. And I have had individuals, if they have the ability to do liquid application, that's great.
It is.
As a seed treatment. But it's one of those things that it's a very exact science. Don't get the seed too wet. Don't, being able to dry it so it's not sticky. It can create problems.
Gummed up
seed
drills
Yeah.
are something that nobody wants to be dealing with.
Yeah, so if you have an understanding, you've done that before and you're very good at it, that's a great way to do it. I have a lot of guys who will auger it in. As they're putting their seed into their seed drill, they're dusting it in. Mixing that through the auger as it goes into those seed drills is another great way to do it.
And a bulking agent that's a food source really helps with
out in that scenario. Yeah, and then I have some growers that are putting it directly into their drills and mixing it up as they put the seed in. So it's it is a great way to get that out. A lot of guys now are using naked seed. A lot of times in that case, we're recommending and having really, really good results with our OP8 product. Doing the Spectrum plus Myco plus the OP8 product or just the OP8 product in-furrow at the time of growing because of the diversity of that microbial community. And as you talked about, creating that rhizosheath on that plant to produce overall plant health, the quicker germination that we see with utilizing this, getting them up out of the ground quicker, getting that plant healthier, photosynthesizing quicker. All of these are critical in creating overall plant health in the different soil environments.
Oh, absolutely. So we've got a wide variety of ways that we can get the material onto the seed, but it's always better to get those organisms present. If we have the option, we should take it, because listening to Dr. White and learning more and more about this rhizophagy cycle, we have to have them. The plant is going to search for them. Whether or not it finds the best players depends on your soil environment or if you're providing them for it. So this is a great way to make sure we're providing those beneficial species, those beneficial organisms that we know the plant loves, needs, and uses right around that seed coating, so that as soon as that germination happens and that new little baby root, that little radicle pops out, it's already got those microbes on and around it. And we see that rhizosheath, we see that dreadlock, we see that, and when we see those, we get so excited. It's so good to see, because we know that plant, it has the best leg up. It has the best chance of performance.
Well, and I've seen that in row crop agriculture before that seed has even emerged out of the soil. Before you even see that, and you dig up that plant, and down in there you have those dreadlocks around that rhizosheath, around that root, down in that soil environment. I mean, it's so exciting when you start to see that. It really is. Now you know you have a biologically active soil, and that plant is functioning more as designed.
It's buffered, it's protected, it's shielded, it's getting the nutrients, it's improving the moisture holding capacity, it's improving gas exchange, all of those things that we want to do as we build aggregate, because that's what we're doing. When we see that rhizosheath, we're building micro aggregate structure on and around that root.
Well, and when we talk about building soil health, not worrying about yield or plant, or the quickest way to build soil health is with a actively functioning, growing, photosynthesizing plant.
Yes.
So if we want to build soil health, this is the best way to do it. It really is.
Maximize photosynthesis, maximize exudates, means more carbon in the ground. More carbon in the ground means we're building soil.
More microbial biomass, everything that we need.
Okay, so we'll switch gears a little bit.
Well, and go into talk about, I mean, this isn't only row crop and in-furrow applications or seed treatment. I mean, we do see the exact same thing happening in root dip a tree and shrub, planting a new orchard, new blueberries, new any time
greenhouse transplant,
strawberries, any of these type scenarios. And most often I talk about it in the orchard scenario where you are receiving these trees where you didn't have it at the point of germination. You're playing catch up. So obviously a lot of times they are dipped in a fungicide, they're sterilizing those roots, and they have to make sure they're not bringing pathogens into a new environment.
So they're starting off with they're starting off a few steps behind on this whole rhizophagy and regenerative and biological approach.
A compromised digestive system.
I would 100% agree.
Yeah. So a lot of times we recommend that root dip, which is, I say, an unbelievable way to get the orchard started. Better than trying to do it through a drip system, or if you can get that microbial community around that root at the time you are planting it and put it right into that ground.
Well, how often do you have access to the roots of a plant? in that sort of environment. Never.
Yeah.
And you never want to again, because if you have exposed roots, you're going to be causing damage. When we have those bare root, when we have those strawberry flats, when we have those plants that we can actually physically coat and surround them with biology, that is when we're creating that fast relationship.
Yeah. So a lot of times, like I say, almond growers, the apple, the pears, the peaches, whatever it might be, the tree fruit, we'll call it.
Ornamentals.
Yeah. They take that, a lot of guys, I usually say a 5-gallon bucket of water. Well, obviously, if we're planting hundreds of trees, a 5-gallon bucket of water doesn't work.
Nope.
We have individuals that take totes, cut them in halves, horse troughs, whatever it might be. Normally, the recommendation is 25 gallons of water to 1 pound of either mycogenesis or rhizogenesis. The mycogenesis is for those organic growers. The rhizogenesis can be used in conventional systems. The only difference between the rhizogenesis and the mycogenesis is the rhizogenesis contains a polymer.
That polymer,
that polymer is not for water holding capability. It's to suck up those like a sponge. It brings in that microbial community. It attaches to that
root gummy
and holds them. It holds them there around that root zone. So that's why the difference between, but they're basically the exact same product. One has a polymer. And then usually, so 1 pound of that with 12.5 ounces of Nutrient or Pepsime G. Nutrient,
just a little bit of food in these scenarios. We've seen burn, we've seen damage when people get a little too excited and they want to give a little bit too much nutrition. We're feeding the microbes. We're not trying to fertilize the plants.
Correct. So if we're looking at putting in a little bit of fish, a quart of fish, a little bit of seaweed to reduce stress.
Oh, seaweed in that situation is fantastic.
But it's microbe food. It is not, that's the only reason we're putting it in there. And if we, you think about it, these are little guys. They don't eat very much. So a little bit of food with the microbes.
Well, the cytogreen's a great application to add.
Yeah, absolutely. The cytogreen is a great product to add to this root dip type scenario. And if we have, more than 25 gallons of water, if we have 100 gallons because we're using a larger tote.
Hold on, we got to do math?
Then we got to do math.
Whoa.
We just combine it by 4.
Okay.
It's pretty
That's
easy, unless
bad.
it's new math. Then that can be trouble.
Fuzzy math, tricky math, who knows?
Yeah. So root dip by far, a lot of guys, what I'll do is I have one guy that has 7 totes that are cut off. He fills them all with trees, has them in this root dip scenario, does that the day before, and then he'll start taking totes out to the field. And as he brings an empty tote back, if it's half full or they've lost a lot of the water, then we come in at a half rate of everything we just talked about. Fill that tote back up with the level of water. We fill it with trees again, and we're just rotating those through that field environment as we're planting. And it's really amazing the difference that we see on something like this.
Absolutely. And that's kind of a good point. We call it a root dip, but the reality, a root soak is going to give those plants better access to more moisture. It's going to rehydrate their cells a bit more. It's going to give those microbes a little bit longer chance. So when we're saying if you can soak them, if all you can do is a quick dip, dunk, and go, that works. It gives us access and again, we want to make sure we've got some of those foods because those are going to help stick some of those microbes to it. But if we can allow those plants to have a few hour or up to
12 hours,
12 hours,
use overnight is what I like to see. And I always tell people, if we're planting over multiple days, with a lot of growers, that scenario, we can't keep that same product in those tubs for 3 or 4 days. It's going to go anaerobic. So after 24 hours, I generally recommend we dump it, we put in new product, we start over again.
Go drench it around those plants you just
put it back out in the field that we just planted. Absolutely.
Yep.
Just don't try and run it through your sprayer because there's generally a lot of debris in there when we come to dipping trees.
Okay, so we have warned people to not do that. So we're good there. No, you don't want a bunch of sand, gunk, grime, root
whatever
bits,
is on the root pieces.
Oh yeah, no, that's a nightmare scenario.
But this isn't only for tree and shrub. I love this in the greenhouse scenario where you're buying greenhouse plants, you're putting out there as a field environment. If we have the ability to treat the seed in the greenhouse, then treat the seed, but still do the root dip before it goes in the field. Adding this to a water wheel is an ideal scenario.
You get food,
everything. I have a lot of large growers that'll dip their flats. They'll have a basin. They'll dip their flats in. With a mixture of the mycogenesis, the nutrient in the food, or the biogenesis that we were just talking about. Just do it the exact same in a root dip there. Take it out to the field, and then, however you're planting, a lot of times we'll have these laid out, a tractor pulling down, and people are popping them into the hole and planting them. great way to reduce the stress. And again, we talk about that seaweed. You know, great plant stress reducer,
Yes.
especially where we're talking about snow plants going into a new environment. There may be a pH shift from the greenhouse to the soil. You know, there may be soil differences, all of these things, root damage as we're putting these guys in. All of the things we can do to reduce stress and have that microbial community there to tell the guy, it's okay. I mean, I do that a lot with you. You stress out, I have to say, it's okay, Steve.
A little pat on the back.
Yeah, a little pat on the back, make you feel
okay.
good.
Yeah. And that's, I mean, that's the beauty. These organisms, they're generating their own plant hormones. They are literally communicating with the plant, and they're changing that stress response. We see that regularly, and you've seen some pretty incredible examples in the field where it helps buffer. If somebody made a little bit of a mistake and maybe they laid their little whips out in a windy, warm environment, and you expect high mortality, if we've done some of this soaking and allowed some of those organisms to establish and start working with the plant, we're offsetting some of that stress. And that's the goal, because transplant time, it is extremely stressful for the plant. So anything we can do to help reduce that stress gives us a much better chance of reducing the number of skips. And that's a really common thing that you see.
Yeah, you know, and the skips are huge. University of Washington put on an article a few years ago in Good Fruit Grower of the cost associated with a skip. Not only the fact of...
It compounds.
Yeah, it compounds. You have to then dig it up. You have to then replant another tree. Now you have a 2-year-old versus a new one. The water is different, the nutrition is different, and then the production yield is different.
Yep.
And just by, when you start to add all those things up, it's just not the cost of the tree.
No.
There's so many more things that compound on the overall losses. And then when you get ready to take out that block of trees,
you've lost productivity.
You've lost another year or two of productivity. So when we start to think about that, it adds up pretty quick. I mean, the numbers were staggering when I started to see that based off a 10% or a 15 or a 20% skip in a field.
Yeah.
Yeah.
It can lead up to tens of hundreds of thousands of dollars pretty quickly. It's incredible. It's kind of shocking and startling sometimes.
So, you know, I mean, one of the questions we get asked a lot of times, we've kind of talked about the in-furrow, we've talked about the seed treatment, we've talked about the root dip, all these spring things that we need to talk about. But, you know, we said we would mention the importance of enzymes because we usually look at that around that soil environment. And I sometimes get the question asked, well, do I really need to utilize the enzymes? You know, I'm putting the biology out there. But if you think about it, those enzymes are critical to biological function and plant function and nutrient availability.
All of those things. Okay, so we take our microbes, we dry them, we put them in a system dormant state, and then we're going to rehydrate them. We're going to try to get them back out there. But what happens when you come out of hibernation? Is a bear hungry? It is starving. So that's the idea behind these enzymes. These are digestive enzymes that are designed to start that process. And the way these enzymes are functioning is, if we look at a cellulose molecule, for example, it is just beta-linked glucose. It's just a big old giant long string of glucose that's linked and linked and linked. Glucose, that's sugar. That is direct food. However, when it is linked together in that form, it's bound up, it is tied up, it is no longer available. So the microbes are going to produce cellulase, which is the enzyme that splits individual glucose molecules off. On a microscopic scale, if we look at even a small chunk of root, a small chunk of leaf that has fallen off and in our soil environment, to a microbe, that's huge. That's like literally eating an elephant. So how do you eat an elephant?
One bite at a time.
So that's what these enzymes are doing. They're specifically targeting bonds, and that's what they're designed to do. They're going after specific bonds between things, like that beta-linked glucose, or we can look at like tricalcium phosphatase, that's breaking apart calcium from phosphorus. It's focusing specific energy on that bond between those molecules. Nitrogen fixation, those triple bonded nitrogens that are linked together, it takes a massive amount of energy to break that apart, and that is built in a very specific structure. So we need to keep in mind what feeds that structure. These enzymes are all built around their core metal ions, their metalloenzymes. You can think about it a whole bunch of different ways, but the reality is they're built around the actual functional group. If you think about these little guys like teeth or like scissors, the actual cutting edge is that trace mineral. Almost all of the time it is a trace mineral, whether it's iron, whether it's zinc, whether it's like we're talking about nitrogen fixation. We always hear people, well, you got to make sure you have your molybdenum levels, right? Well, the reason for that is that nitrogenase enzyme, the specific functional area of that enzyme is that molybdenum. That's where the magnifying glass kind of occurs. And that's kind of what's happening is nature's amplifying the energy to break that bond. And the way it's doing that is through that specific site, that specific structure. So we need to make sure we focus on all those trace minerals and getting a wide diversity out there. So the microbes, the plant, all of these different living systems can create this wide diversity of enzymes because they are what is driving every one of these biological functions. So we need to get that out. And one of the easiest ways we use is anything from the ocean. If we've got something from the ocean, or if we've got a good, very micronized rock flour, rock dust that has a wide diversity of trace minerals, that's a great way to make sure we're getting those molecules, those trace minerals out there so those enzymes can be built. And when we do this, okay, back to the whole idea of, breaking it apart to make that food available. Okay, so what that has then done, if we look at that glucose that's been split off from that cellulase, or from that cellulose molecule by that cellulase, now the microbe has access to sugar. And it's that sugar that's going to feed it. So we've got these hibernating microbes that are very, very hungry. If we pre-apply and pre-digest some of that food for them with our enzymes, the microbe wakes up, and then rather than having to generate that enzyme itself to start that digestion process, it's already happening. So we're already feeding those microbes.
And the amazing thing about these enzymes, I give the example that they're like an end wrench. If it's a 7/16 inch wrench and I need it and I tighten the nut in the shop, then I put that back in my toolbox.
You can use it again?
I can use it again. And that's what these enzymes, once they break that bond, they release it, they go find another one, and they break another bond. And they just continually do that within that soil environment. But if I don't have 7/16 inch wrench,
you don't have access.
I can't do that function.
You can't break that. You can't work on that. If you don't have the trace mineral to create that enzyme, you can't break that bond. It does not happen.
And that's what's pretty cool about it. So we talked a little bit about the enzymes, the importance of enzymes, but really to get this whole process started, as we've seeded our field, early on application. Now we want to start to talk a little bit about feeding those microbes, keeping them active as possible, and a lot of times that comes to exudates that's in that soil environment and foliar nutrition.
Yeah, and that's where we really need to focus. So we've got the plant happy, it's got a beautiful rhizosheath, the rhizophagy is happening, our roots are happy. So how do we make sure we're keeping that photosynthesis, those exudates at optimum? And that's exactly it. And we're doing that with course corrections. If you're trying to do all of your fertility through foliar nutrition, it's going to be expensive, it's going to be difficult, and it's easy to make mistakes because we're overemphasizing concentrations sometimes. So when we're talking foliar nutrition, I'm going to say it, I know everybody's sick of it, but test, don't guess. We need a driven, targeted decision. We need to know what that plant needs in order to make the most appropriate application. And that's where we come in with this foliar nutrition. And that's one of the ways that we can get maximum performance out of our plants.
And a lot of times, because growers will always ask me, well, how can a pint of this or a quart of that make any difference in overall plant health? And what we look at it, I guess the way I look at it is it's through communication and those exudates going down into that soil environment and going back into what we talk with that rhizophagy cycle. But in order for that to happen, that's a communication. We have to have actively functioning plant photosynthesizing, putting that nutrition down in to get that process or keep that process going. And that's what we really see with a good foliar application. We look at our PZ 1000 or Micro 5000 or Micro 5000 organic. Those are all designed with everything we've talked about in the soil environment up to this point happens on the phyllosphere of that plant, happens on that leaf structure. So we can't microbially ignore it. The phyllosphere. And so these phyllosphere organisms are helping make phosphorus available, potassium available, all these trace minerals available. They're sticking them to the leaf of that plant. They're creating their little home, their biofilms on that leaf structure. So they have an environment because they need those foods. And that's not only on the outside of the plant, that's on the outside, inside the leaf, the endophytic. All of this happens on that leaf structure of the plant, helping making nutrition available. And then we add that plant sap analysis and we know we're a little bit deficient on manganese or we're a little deficient in calcium. And we put those foliars on there, we put them, it's an IV to that plant. We put it right into that plant. It then puts that communication down to that soil environment and said, I need more calcium. I need more manganese. You guys find it for me.
And then it allows that plant to do it. And we have a few other kind of more targeted products. The calcium 24 is one. It's not organic, so not everybody can use it. However, we have a lot of, I'd say some of the nut guys, the nut producer, the almond industry, or almond, uses a large quantity of it. And we have a lot of apple producers as well that are utilizing it because we know when we're struggling with calcium, calcium is difficult. If our soil is not supplementing the calcium levels that that plant needs, we need to get it in. The hard part is calcium is not mobile. The beauty of calcium 24 is it's an amino acid chelate, allowing that charge, that calcium 2+ charge, to be masked. It's basically coated, surrounded, and it's going to be transported around because amino acids are needed by plants. The plant wants that amino acid, and having it in that form allows it to be translocated anywhere in the plant. And it helps decrease things like bitter pit. We see issues that are associated with calcium start to decline when we have adequate levels of calcium.
Blossom end rot. I mean, the list goes on.
It does.
And, you know, and one of the things about it, it's not inexpensive, okay?
No.
But it is, if you want to get calcium into your plant, it's a great way to do it. And when we say it's not inexpensive, what is the amount of crop loss when you leave bins of apples or tomatoes or whatever into the field because they're rotting because we don't have sufficient calcium in that plant for that cell division and all those things to function. Now, when you think about it that way, well, that's pretty cheap.
It is.
You know, compared to calciums that you put out there, that you put it out, you do a plant sap analysis, and you notice that nothing changed. You know, so I always say, utilize those tools to make sure that what you're putting out there in the field is actually, we're seeing a plant response from that in the positive direction that we want to see. You know, and you know, when it comes down, Micro 5000 Organic is probably one of our highest used products. From the idea, it is one product that all growers can use because it is organic certified, so it's very easy to use. A lot of the conventional guys are using the Micro 5000 regular or the PZ 1000. We cannot use those in organic certification. A lot of times my recommendation is the 5000 early when we're in the vegetative state, and then PZ 1000 afterwards when we go into the reproductive.
Higher potassium load.
Yeah, that's kind of the way those two products are built. When we see some crops like strawberries or tomatoes, which shift vegetative, reproductive, it's nice to ship back and forth between PZ 1000 and Micro 5000. But for most scenarios, early on Micro 5000, then follow up with PZ 1000. We see a lot in the fruit industry for the conventional guys. They'll be using pre-bud, bud swell,
Micro 5000,
after petal drop, maybe Micro 5000, and then shift to the PZ 1000 once we start to create that fruit, that nut, whatever that scenario might be. And then, like I say, a lot of the guys are just using the Micro 5000 Organic. They don't have to worry about which one I use. They just use it from bud swell all the way post harvest. You know, after they're done picking, they give that plant a water irrigation cycle. They put their soil inoculum out there, and then they come in with another foliar. I mean, that get some energy back in that plant at the end of harvest. That's really what we start to talk about.
And those are nutrition focused. We also have a lot of people that are using more microbes on the leaf. Dennis was talking about how important that phylobiome is and how we see those benefits. A lot of that same communication, those microbes that are on the phylobiome that are creating the phytohormones for the plant, communicating with the plant, we can do all these same things. With endophytic nitrogen fixers, we can have a spray of beneficial microbes that has these endophytic nitrogen fixers go out as a foliar, establish with the plant, and now we have nitrogen fixation happening throughout the plant. We've already treated the root, now we're treating the leaves. We're getting that nitrogen fixation happening everywhere. And as Dennis mentioned, if we have phosphate solubilizers, phosphorus is another one that is difficult to get into the plant. If we have organisms that we're creating this phylobiome around that leaf that contains these beneficial organisms, it's increasing the uptake of the other foliar products that we're using. We're increasing that plant health. So utilizing a little bit of the spectrum and utilizing some of those other microbial products that are exciting and fun to play with to make sure we have a good, healthy phylobiome can be critical.
And I'm seeing that like in the winter wheat. Coming out in the springtime, that early application of a foliar application, right? Early tillering is when we want to put that out there. I call it, we put energy back into that plant. It's coming out of dormancy, like you said, the bear, we get that food in there, we get energy in there. It's like eating a Snickers bar, Marcia. And getting it up and running and off it goes, and we get that exudates, and that's what starts the whole process. So, with that, a lot of times we talk about in-field applications, the different applications throughout the growing season. We see this a lot in the fruit growers. We see it a lot in the row crop industry. Is we've come in with our seed treatment, our in-furrow application. Then we're pulling a plant sap analysis, and we notice that we're not getting the phosphorus that we need. We're low on calcium. We need more nitrogen. Then a lot of times we can come in with an additional application, soil applied, ground applied, through your pivot, through your drip irrigation system. It can be ground applied through a ground rig if you're dryland farming, of a specific product based off of function. That's where we talk about the DS. If we have high sodium or high salt within that plant tissue, if we don't have enough nitrogen, the spectrum NFB in there for the nitrogen fixation. If we're lacking phosphorus, the spectrum PSB. They're all designed based off of function. They have that foundation of that microbial community, that diversity of everybody's there, with an emphasis on a specific function. That's really what it comes down to.
Well, and starting off, like we talked about, one of the most commonly used is the spectrum plus myco. So if we can get those PGPR plus all those mycorrhizae, if we can get the mycorrhizal fungi attached with the plant, and then we come back, those mycorrhizae generally take 3-4 weeks according to research. So if we come back at that 3-4 week time with our spectrum PSB, now we have the mycorrhizae that are great at translocating water, as well as many of them are great at moving phosphorus to the plant. So then we come in and we apply that spectrum PSB to the soil, increasing the phosphorus availability to the soil. Now guess what? You've got all those little microscopic straws that are the mycorrhizal fungi that can help move all of that to that plant. Because these microscopic bacteria, they're the ones that are generally down there, and they're in the trenches. They're the ones with little tiny picks and miner hats that are actually creating the enzymes and the acids that are extracting a lot of that phosphorus so it can be translocated to the plant through those mycorrhizae.
Yeah, so with that, I know that was a lot of information. We've talked about a lot of different things. We've talked about from basically seed treatment, in-furrow to root dip, the Foliar to enzymes, all the way through. If you have any questions about anything we talked about, if any one of these pieces fit into your puzzle, that missing link that you're trying to find for your agriculture, for your growing practices, give us a call.
Reach out.
Reach out. We'll be more than happy to be honest and answer any questions you might have about any of our products. So thanks for joining us. Again, I'm Dennis.
I'm Steve. Thank you everybody for joining us for the Natural Growing Through Biology podcast.